Welcome back to the deep dive. If you've ever wondered how the trillions of cells in your body manage to act like a coordinated orchestra instead of just a, random soup of organelles, this deep dive is for you.
It's a great question.
Today, we are cracking the molecular language of life itself. We're talking messengers and receptors, focusing on how cells receive, amplify, and maybe most crucially, how they manage to shut those signals down.
That's right. Our mission today is to synthesize the foundational mechanics of cellular communication, which can get really dense with acronyms like GPCR and RTK.
Oh, absolutely.
So we're translating these into clear, usable knowledge. I mean, fundamentally, multicellular life is a symphony. And that symphony relies entirely on cells producing and interpreting signals, whether that's a quick chemical release or, just displaying molecules on their surface.
Okay, let's unpack this by starting with maybe the most basic variable in signaling distance. How far does the message need to go? When we look at this, there's a whole spectrum. At the absolute shortest distance possible, you need physical contact between two cells. That's what we call juxtacrine signaling.
And just a tiny step up from that is a really clever move cells use to reinforce their own behavior. If a cell releases a signal that then acts directly back on the same cell that sent it out, that's autocrine signaling.
It's like sending a reminder text to yourself.
Exactly like that. It's a feedback loop.
And then you have the long-haul delivery service. Endocrine signaling, this is the world of hormones.
Right. These are signals made by endocrine tissues. They get dumped directly into the bloodstream, and they can travel all over the body to act on really distant targets.
Yeah.
It's the master regulatory system.
And when we look at these endocrine hormones, all the key characteristics hold true, right? They're made by specific glands, they travel in the blood, and their lifespan can be, all over the place, from seconds to hours.
That's right. But the delivery system is only half the battle. The real biological challenge is reception. How efficiently does a cell even hear that signal? And this brings us to the math of it all. The dissociation constant, or text KDD.
And this is where the strategy of cellular design just really shines. Experimentally, the TextD is defined as the concentration of free ligand you need for exactly 50% of your receptors to be bound. That's the saturation point.
And that number is everything. The TextD2 is the measure of the receptor's affinity for its ligand. And crucially, and this trips people up sometimes, a low TextD means a very high affinity.
So you need less messenger to get the job done.
Exactly. You need less of it to hit that 50% binding mark, which means the receptor is incredibly sensitive. And that sensitivity dictates how responsive a cell is. For a signaling system to even work, its TextD has to fall right within the normal physiological range of that ligand.
It has to be in the sweet spot.
It has to be. If the signal ranges from, say, less than $10 9 along dollars, the receptor needs a text stack right in there to make sure it only fires when the signal is actually relevant.
That makes perfect sense. The receptor is tailored to the concentrations it expects to see. But, okay, the job isn't done when the signal arrives. The cell also needs an exit ramp, right? It can't stay on forever.
Absolutely not. That would be a disaster. The mechanism for protecting itself from prolonged exposure is called receptor desensitization. It's like the cell turning down the volume on a song that's been playing for too long.
So how does it do that?
A couple ways. It can physically remove the receptor from the surface through endocytosis, basically just pulling it inside the cell, or it can use biochemical tricks like phosphorylation to temporarily shut the receptor off while it's still on the membrane.
So just to be clear, simply destroying the ligand floating around outside, that helps lower the signal, but that's not actually receptor desensitization itself.
That's a key distinction. Exactly. One is cleaning up the environment, the other is changing the receiver.
Got it. And that leads us right into the first major class of these receivers, the G protein-coupled receptors, or GPCRs. These are the giants of cell surface signaling.
They really are. They're involved in everything from your sense of vision to your sense of smell.
And what's so fascinating is their structure. They all share this characteristic design, a single polypeptide chain that snakes back and forth through the membrane 7 times.
Yep, the 7 transmembrane alpha helices.
Yeah.
You see it in the beta-adrenergic receptor for epinephrine. You see it in olfactory receptors, opioid receptors.
But not everything uses this structure.
No, and it's important to remember that. Receptors for things like steroid hormones, which are small and can just slip through the membrane, they're not GPCRs, they're usually intracellular. They're waiting inside the cytoplasm for the hormone to arrive.
Let's focus on that GPCR mechanism, though. The moment the ligand binds, it kicks this whole machine into gear. It activates the heterotrimeric G protein, that text alpha beta gamma that's just waiting underneath. Can you walk us through that cycle? It's like a perfect little switch.
It is an elegant switch. So when the GPCR is activated, the text alpha subunit literally drops its old GDP molecule and picks up a fresh, high-energy GTP.
Okay.
That GTP binding changes its shape, forcing the text alpha GTP unit to break away from its text beta gamma partners. And now you have two active signaling pieces, text alpha and text beta, that can go off and do different jobs.
And we classify these, right? Like zeros for stimulating and several dollars for inhibiting.
Exactly. But the most important part is that built-in timer you mentioned. The text alpha subunit is itself a GTPase.
So it turns itself off.
It turns itself off. It eventually hydrolyzes that GTP back to GDP, which makes it snap back together with the text beta gamma complex, signal over, system reset.
And this quick self-resetting cycle is why GPCRs are perfect for fast signals, like an adrenaline rush from epinephrine.
Precisely. In that case, the active DG dollar subunit goes and activates an enzyme called adenyl cyclase, which immediately starts cranking out our major second messenger in this pathway, cyclic adenosine monophosphate, or CAMB.
And CANMP is the real messenger here. So what does it target?
It targets protein kinase A, or PKA, but here's another critical detail. CANMP doesn't just bump into PKA to turn it on. It activates PKA by binding directly to PKA's regulatory subunits.
Ah, I see. So PKA is an assembly of 4 subunits.
Correct. 2 catalytic, 2 regulatory. The regulatory ones are like a safety lock. When CAMP binds to them, the lock comes off, and the 2 active catalytic subunits are set free to go and phosphorylate their targets.
So the cascade is crystal clear then. Ligand binding, G protein activation, adenyl cyclase activation, a flood of CANMP, and then PKA activation.
And the downstream effects are enormous. When epinephrine hits a liver cell, PKA is the protein that takes direct control of glycogen metabolism. It rapidly triggers glycogen breakdown into glucose while also shutting down the enzymes that make new glycogen.
An instant energy boost.
Exactly.
Now let's look at what happens when that perfect timer fails, because this is where you see just how fragile this whole system is. The story of bacterial toxins is, well, it's a terrifying lesson in molecular warfare.
It really is. Take cholera toxin. It targets that $L subunit and chemically modifies it so it cannot hydrolyze GTP. The timer is broken.
So it's permanently stuck in the on position.
Permanently.
Yeah.
Which means adenyl cyclase runs nonstop, churning out insane amounts of CAMP. In your intestinal cells, this causes a massive, relentless secretion of fluid, and that's why cholera leads to such severe, life-threatening dehydration.
Wow. And then you have pertussis toxin. It's similar, but different, right?
Yeah, it's a related mechanism, but a different target.
Yeah.
Pertussis toxin goes after the inhibitory dollar subunit. It prevents it from doing its job, which is to inhibit adenyl cyclase. So the break line is cut. The result is the same. Adenyl cyclase runs wild, ANP accumulates, but the way it gets there is different.
That's profound. The off switches are just as critical as the on switches. Okay, moving on from KMP, let's talk about the universal language that ties all these pathways together.
Cool.
Phosphorylation. At the most fundamental level, the job of kinases and phosphatases is to directly turn proteins on and off just by adding or removing a phosphate group. It's the cell's binary code.
Absolutely. Kinases add the phosphate, phosphatases strip it off. That dynamic balance controls the activity of almost every protein in the cell.
And speaking of activity, let's look at our next major second messenger, which often works with GPCRs, calcium TXDA2 plus, PEACE. This pathway usually starts when a G protein activates an enzyme called phospholipase C.
Yes, and phospholipase C is like a molecular pair of scissors. It finds a specific lipid in the membrane, TXPIP2, and it cleaves it into 2 separate second messengers at the same time. You get TXIP33, which is water-soluble, and DG, which stays behind in the membrane.
And TXIP3 is the trigger for the internal calcium release. So the TXIP3 receptors, they're on the endoplasmic reticulum, the ER, right?
Exactly. There are gated channels right on the ER membrane. When TXIP3 binds, it opens those channels, and calcium, which has been stored at high concentrations inside the ER, just floods out into the cytoplasm.
You have to appreciate the baseline here, too. The cell works incredibly hard to keep the resting cytosolic calcium concentration ridiculously low, something like near $1.
It's an obsession for the cell. It uses powerful calcium ATPase pumps and TEX2 plus exchangers to constantly pump calcium out or back into the ER. So when those TXIP3 gates open, the concentration can spike 100-fold almost instantly. It's a huge, sudden signal.
And once that calcium is out, it needs a handler. It doesn't just float around. That handler is the protein calmodulin.
Calmodulin is perfectly engineered for this. It's designed to bind two TXGD plus TOS-ILers at each of its two ends. So 4 in total. But here's the key. It does not bind them irreversibly.
It has to be able to let them go.
It has to. Its
Wow.
whole job is to bind calcium when the concentration spikes, change its shape to activate other proteins, and then let go of the calcium just as quickly when the pumps restore the baseline.
Now, how do researchers even study this? It's happening so fast.
With some incredibly clever tools. To measure these calcium fluxes in real time, in a living cell, scientists can use synthetic proteins called chameleons.
And what if you're in the lab and you just want to force the system? You want to cause a calcium release yourself?
Then you'd use a calcium ionophore, something like A23187. It's a small molecule that basically punches holes in the membrane and lets the calcium flow through, bypassing the whole receptor system.
You see how all these pieces fit together when you look at something like nitric oxide signaling? For NO to dilate a cardiac artery, you need guanylate cyclase, you need CGMP, you need protein kinase G, you need calmodulin. So an inhibitor of any of those steps would block the whole process.
It's a beautiful, intricate cascade.
Okay, let's switch gears from the GPCRs, which are all about speed, to the receptor tyrosine kinases, the RTKs. These are the receptors for long-term strategy, right? Growth, proliferation, differentiation.
That's the perfect way to frame it. RTKs are stimulated by key growth factors, things like EGF, PDGF, FGF, and this is a big one, insulin.
And here's the key exception that always seems to come up on tests.
Oh yeah, this is the one that trips people up. Transforming growth factor beta, PGF-V beta to 4. It sounds like a growth factor, but it activates a serine threonine kinase receptor, not a tyrosine kinase receptor. And that distinction is a fundamental partition in cell signaling.
So mechanically, how do RTKs work? They're usually single chains sitting in the membrane.
Right, they're just waiting. When the ligand binds, it causes them to pair up, they form dimers. And that dimerization is the key. It brings their internal tyrosine kinase domains close enough to phosphorylate each other.
That's the autophosphorylation step.
That's it. The receptor lights itself up on specific tyrosine residues, and those phosphorylated tyrosines become docking sites for dozens of downstream signaling proteins.
So if we had that hypothetical alien factor, XFF, and we wanted to prove it acted like EGF, finding a tyrosine kinase inhibitor that stops its signal would be the smoking gun.
Absolutely. That would be your proof.
Once that RTK is lit up, it sets off the legendary RAS cascade, which is so central to cell growth and, when it goes wrong, to cancer.
It all starts with the activated RTK recruiting an adapter protein called GRB2. GRB2 then grabs the real switch operator, a protein called SOS, which is a GEF.
The guanine nucleotide exchange factor.
And its job is to activate the small monomeric G protein, ROS. So SOS basically forces ROS to drop its inactive GDP and pick up a fresh GDP. Now ROS is active.
An active ROS GTP kicks off the MA pinase cascade. ROS activates RAF, RAF activates MEK, and MEK activates the MEPKs, the mitogen-activated protein kinase.
And those final MEPKs are the ones that head into the nucleus to phosphorylate transcription factors that turn on genes for cell growth and division.
And just like the GPCR system, there has to be an off switch.
There has to be. In
Yeah.
this case, it's GAP, a GDPase activating protein.
Yeah.
GP helps ROS hydrolyze its GDP back to GDP, shutting it down. And just to be clear, ROS is a G protein, but it absolutely does not bind steroid hormones.
The genetic proof for this pathway is just beautiful. The work in Drosophila flies.
Oh, the 7-less experiments. It's classic molecular genetics. The 7-less RTK is required to make the R7 photoreceptor cell in the fly's eye. If you have a broken SEV receptor, you lose that cell.
But here's the kicker. If you take that fly with the broken receptor and you artificially express a permanently activated RAS protein,
The R7 cell develops perfectly fine. It completely bypasses the need for the receptor. And that one experiment proved that the 7-less RTK's normal job is simply to turn on RAS.
these insights are so powerful when you look at human diseases too. You see dominant negative mutations, where a broken receptor pairs with a normal one and poisons the whole complex.
Right, it makes the cell deaf to the signal. And the opposite is a constitutively active mutation, where the receptor is stuck on even with no ligand. A classic example is the FGFR3 receptor mutation that causes achondroplasia, a form of dwarfism, because the growth-regulating receptor is constantly signaling stop growing.
We can't finish without looking at how these systems integrate. Let's touch on insulin signaling. It uses an RTK to manage glucose.
Right. So insulin binds, the receptor activates, and it phosphorylates a docking protein called IRS1. IRS1 then turns on PI3 kinase, or PI3K.
And PI3K converts the membrane lipid text PIP2 into text PIP3.
That text PIP3 signal then activates ACT, also called protein kinase B. And ACT is what drives the metabolic outcomes. It gets the GLUT4 glucose transporters to move to the cell surface to pull in glucose, and it ramps up glycogen synthesis.
And what's the off switch here? There's always an off switch.
The key apposition here is an enzyme called PT10. PT10 is a phosphatase that specifically reverses what PI3K does. It converts text PIP33 back to text PIP2. So PT10 is an inhibitor. It absolutely does not promote insulin signaling.
It's all about that balance. And of course, there's signaling crosstalk where all these pathways intersect and influence each other.
It's a massively complex web. And to handle that complexity, the cell uses brilliant organization, like scaffolding complexes. The ST5 complex in yeast mating is a great example.
Oh.
It physically grabs all the kinases in the MEPK, cascade RAF, MEK, and EPK and holds them together, ensuring the signal is fast, accurate, and doesn't get lost.
We have covered a lot of ground today, from the text for G that dictates sensitivity, to the molecular warfare of bacterial toxins, to the different strategies of GPCRs and RTKs.
At the end of the day, these are the essential tools cells use to interpret the world around them. And that precision is everything. As you said, a tiny molecular change, a single mutation making a receptor constitutively active, a toxin locking up a TEX-alpha subunit can derail the entire system and lead to disease.
Right. We can now look at some unknown factor, like that alien XFF from the source material. And just by seeing what kind of inhibitor stops it, we can tell if it's acting like a growth factor on an RTK or maybe a hormone on a GPCR.
So, as you integrate this all into your knowledge base, here's a final thought for you to chew on. We've seen that signaling is a complex network, this thing called crosstalk. So consider this. A cell receives two powerful and often conflicting signals at the exact same time. Let's say a stress signal, like epinephrine, screaming for energy breakdown via the CAMP path, and a growth signal, like EGF, demanding energy storage and proliferation via the RTKR's path.
That's a great question.
How does the cell biochemically decide which urgent message to prioritize? And what are the molecular safety mechanisms that prevent these two incredibly powerful and often opposing pathways from completely overriding one another? Something to consider until our next deep dive.